All good. So good morning, everyone. Thanks for coming. So for the second week in a row, we're having a CAML Rheumatology series. This week, we have Dr. Mojca Frank-Bertoncelj, I hope I pronounce it right, who comes from Germany. So Dr. Frank-Bertoncelj is an accomplished researcher and educator in the field of experimental rheumatology. She obtained her medical degree from the Faculty of Medicine at the University of Ljubljana in Slovenia in 2005. She then went on to receive a PhD in biomedicine from the same institution in 2010. She did her postdoctoral research and served as a research associate at the University Hospital Zurich and University Medical Center Ljubljana before becoming a junior group leader at the Center of Experimental Rheumatology at the University Hospital of Zurich. Currently, she leads the team Tissue Protective Factors in Autoimmune Diseases at the BioMed X Institute in Heidelberg in Germany. The team aims to understand the molecular mechanisms that promote pro-resolution responses in immune and stromal cells. It utilizes the human synovium as model systems to characterize the pro-resolving phenotypes of macrophages in inflammatory arthritis, aiming to identify new drug targets. Dr. Frank-Bertoncelj has supervised numerous PhD and master theses, postdoctoral research projects, and research projects of interns and stromal cells. Oh, okay. And of intern and undergraduate students in experimental rheumatology at institutions in Slovenia, Switzerland, and Germany. She has secured over 2 million euros in research funding as a lead investigator, and she has published more than 50 publications in peer-reviewed journals with an H index of 23 and 1,700 citations. In addition to her research, Dr. Frank-Bertoncelj is an active member of various scientific societies and serves as a member of the editorial board of rheumatology. She's an esteemed and well-respected expert in the field of experimental rheumatology, and we are honored to have her speak here at this conference today. Mojca, thank you.
Yeah, and today I would look a bit into back in the past, what I've done, what our team did, and also what other researchers did in the field of single cell omics and spatial omics to reveal the signatures in the human synovium in inflammatory arthritis. So the roadmap of today's talk. First of all, I would like to briefly touch the precision medicine gap in rheumatoid arthritis, mainly from a translational perspective. And then I would come to the clinical and scientific breakthroughs that would hopefully help us to close this precision medicine gap in RA. And I would come then to single cell omics studies that allowed us to gain significant insights in synovial tissue composition and heterogeneity between patients. And finally, I would also give a perspective how single-cell omics has taught us about synovial pathology and what are the new translation potentials for drug discovery in arthritis. And with this, I would come to so what is precision medicine in complex human diseases? It allows us to prescribe the right drug to the right patient based on knowing the underlying molecular and cellular pathobiology. What you can see here. So what do we need to know to be able to do precision medicine? We need to understand the disease pathobiology at the level of a molecule, a cell, and a pathway. And we can then discover or repurpose the right targeting drugs that target specifically either the molecule, cells, or pathway. Furthermore, we need to understand disease heterogeneity. So in which patients, the specific cell types, molecules, or pathways are active and drive disease. And precision medicine has reached a significant success in oncology. And this was mainly catalyzed by the early access to tumor tissues. So what do we know at the moment about cancer? We know very well the oncogenesis process, the cancer pathobiology. So we know the genes, molecules, and cell types and pathways. We also understand very well the heterogeneity of a tumor. And this both together drives drug discovery. And the result is that there are many, many drugs, as you can see here, that can be targetedly prescribed to the right patient. And this has driven the favorable outcomes in cancer. And now coming to chronic inflammatory arthritis, including RA. So chronic inflammatory arthritis present a group of chronic inflammatory diseases that primarily affect the synovial joint. And rheumatoid arthritis is one of the most common one and most destructive ones. So you can see here, there are the feature of rheumatoid arthritis. You can see the joint pain and patients and swelling. And if not treated, this disease brings about very terminal and perturbed joint state with bone and cartilage damage, significant morbidity, decreased life quality, and also premature mortality. Rheumatoid arthritis is now known as being a heterogeneous disease, and there is a fundamental pathogenic role of two major compartments of the immune cells and also joint resident stromal cells. Rheumatoid arthritis has been a long time at the frontier of arthritis and also rheumatic diseases research and drug discovery. And this means that we have today effective drugs that target particularly the immune system and the inflammatory process. So what has been often seen before is not anymore present today. So most patients can reach this state of remission or at least of decreased disease activity. This happens in approximately 50% of patients. And this is driven mainly by our choice of synthetic biology and biologic targeted therapies. So there is an array of targeted therapies in rheumatoid arthritis. However, we don't know what specifically these drugs target and which patients will benefit most from them. So despite the therapeutic revolution that has been seen in RA over the last decades, the treatment choices in RA are still chosen based on a trial and error approach. And what we see, despite the good treatments, we see an inadequate response to single drugs in 30 to 40% of patients. And there is a refractory RA that occurs in 5 to 20% of patients. And these patients see an inadequate response from multiple drugs. And what we know now is that inadequate understanding of RA biology has been actually the stumbling block in precision medicine advancement. So one of the core things was a limited access to synovial tissue across disease stages. We have underestimated significantly the RA complexity. There are no drugs that target the stromal cell compartment. And we have a limited understanding of RA heterogeneity, which also consequently leads to no biomarkers that would predict, for example, disease severity or treatment response. However, the picture is not great because there were several breakthroughs that will or are already facilitating the development of precision medicine in RA. So the major thing that happened was actually the development of ultrasound-guided synovial biopsy. And this allows us now access to clinical samples within a joint, so the human synovium as a principal disease site. We can build now the large patient cohorts, and there were tissue processing protocols developed over the recent years. And this was paralleled with the omics and computational methods revolution, particularly looking at a single cell level and also of cell organization in space. Finally, there was a boost in development of 3D tissue engineering methods. And this was facilitated by stem cell differentiation applications, extracellular matrix biology, and advancement in bioengineering. And just a few months ago, so in January 2023, the FDA released a statement that drug testing is not necessarily needed on animals before coming to the human trials. And when you look now the span of these developments, it becomes clear that if we want the advancement, we need international, multi-center, and interdisciplinary partnership that can bring together all these different types of knowledge. And what the model that appeared within this partnership is mainly the public-private partnerships and also academic industry consortia. And particularly in the field of RA, there were two large consortia as example that developed over the recent years. And one of them is Accelerating Medicines Partnership, which is a public-private partnership. And the aim of the consortium is to transform development of new diagnostics and treatments across many diseases. And there was a significant progress, particularly in rheumatoid arthritis. Furthermore, as exemplified by Matura Partnership, again, an academic industry partnership, which wants to identify and test predictive biomarkers in response to biologics in RA. So, having now established the major lines that allow our advancement, I want to give you a closer look to the specific breakthroughs that might facilitate the advancement of precision medicine in RA. So what is the human synovium? This is the inner soft tissue layer of the joint capsule. So here you see the joint capsule that encloses the joint space. And this is the outer fibrous part, and this orange layer is the inner synovial tissue part. And when we look into the synovial tissue, what we see that actually it is composed of two compartments. This is the synovial lining that faces the synovial cavity and is composed of a layer of synovial macrophages and synovial lining fibroblasts. So these are synovial resident cells. And then we have this interstitial tissue, which is interspersed with fibroblasts, macrophages, and blood vessels in a healthy synovium. And this is basically how the healthy synovium looks like. So here you have the joint cavity, the layer of macrophages, and then the fibroblasts interspersed sublining. Furthermore, when you look in RA, this tissue becomes very tumor-like. So the lining layer is disaggregated and disordered, and you can see a heavy proliferation of immune cells and also of these stromal cells and invasion of many immune cells from the bloodstream. And of course, the development, as I previously said, of ultrasound-guided synovial biopsy has allowed us to really look to get the synovium and look at it from different perspectives. The ultrasound-guided synovial biopsy, as probably most of you know, is a well-tolerated and reliable and minimally invasive procedure that allows us to obtain high-quality synovial tissues from both small and large joints of patients with arthritis. And the minimal criteria that exist to get really good and representative tissues are to get the 6 to 8 samples, and that the synovium that we get contains synovial lining and sublining. And this minimality of 6 to 8 samples pertains to the heterogeneous distribution of the inflammatory process within the joint. So the aim of synovial biopsy in the clinical space is to facilitate early diagnosis, prediction of disease severity, and also prediction of response to therapy. So to facilitate development of precision medicine. And in translational space, having access to synovium allows us to really study the complexity of RA pathobiology, heterogeneity, and also identify new molecular and cellular targets to drive drug discovery. And they are, of course, connected and facilitate each other. So there were several trials to develop synovial tissue dissociation protocol that would allow us to gain really highly viable and representative cell types. And one of the methods was developed by AMP consortium, so Accelerating Medicines Partnership. And this method for synovial tissue dissociations allows analysis of the cells released from viably preserved synovial tissue that were frozen. So it allows the multicentric collection of synovial tissues, and this was done across several states in US and also in England. And then these samples are frozen and then sent to the central processing site. And then the tissue is dissociated in a combined mechanical and enzymatic dissociation to get single cells. So this allows for a centralized, standardized tissue processing, and these cells were used to perform different types of single cell omics on sorted synovial cells, also on predefined synovial cell types. Furthermore, the second protocol was developed by our team in Zurich, and this is a single center perspective collection of small synovial biopsies. And in our case, cells were dissociated from fresh tissue. And we refined the primary protocol for cryopreserved tissue for allowing for cell enrichment and high viability. And of course, we performed single cell RNA sequencing on these unsorted synovial cells, including synovial neutrophils. So finally, the single cell multi-omics, if you look into human body, it is composed of billions of cells of heterogeneous types and also functional states. And unimodal single cell omics, so looking at, for example, the gene expression, allows us now to define cell types and their functional states. It allows us to understand cell plasticity upon perturbation. For example, if we give and treat the cells with different drugs. And it allows us to understand cell differentiation processes and reprogramming trajectories. And in the recent years, there was a significant development of multimodal single cell and spatial omics that touch different levels of regulation in a cell. So we can look up into DNA, the epigenome, the transcriptome, and a proteome of single cells. And we can also understand their spatial distribution in the tissue. And this phenomenal development of single cell and spatial multiomics and computational technologies significantly advanced our understanding of complex human tissues and their pathobiology and heterogeneity in health and disease. And what we know now is that there are unique and shared cell types that's across pathologies and across tissues. And this can significantly push the drug discovery and repurposing. So having this milestone reached, I would like to give you now a perspective of what these single cell multi-omic studies teach us about synovial tissue composition and heterogeneity. And coming back to one of the primary AMP studies. So what the researchers from AMP consortium did here is that they would collect the synovial tissue from different sites. And this synovial tissue was used for histology, but was also disaggregated into single cells. And the single cells were then analyzed by different modalities, including flow cytometry, mass cytometry, and RNA sequencing, and also they were sorted for single cell RNA sequencing. So this was really an integrative look to each single cell in the human synovium. And what could they see is that human synovium is containing the different cell types, so fibroblasts, monocytes and macrophages, different types of B cells and different types of T cells. These were sorted synovial fibroblasts, monocytes, macrophages, B and T cells. They gathered more than 5,000 single cell RNA sequencing profiles and could define 18 different distinct synovial cell populations. And when you look at these distinct synovial cell populations across patients and leukocyte poor osteoarthritis and rheumatoid arthritis versus leukocyte-rich rheumatoid arthritis, you can see a significant variability across all the four analyzed cell types. So then integrating these different modalities, the researchers for AMP consortium could see and identify the cell states that are expanded specifically in leukocyte-rich RA synovial tissue. And these are just a list of specific sublining, fibroblast subtype, pro-inflammatory monocytes, autoimmune associated B cells, and peripheral helper and follicular helper T cells. In contrast, they could see that in osteoarthritis synovia, the lining synovial fibroblasts are expanded together with NUPR, tissue resident macrophage. What does this mean? So these cell types that are specifically enriched in a RA versus OA can potentially be the next cellular therapeutic targets for both diseases. Furthermore, we in our team in Zurich performed and created the single cell atlas of the fresh human synovium in inflammatory arthritis. And you can see here that we used different types of inflammatory arthritis. Most of patients were derived from, had RA, some had psoriatic arthritis, a few patients with spondyloarthritis, and a few patients with undifferentiated arthritis. And these were freshly dissociated synovial cells. They were unsorted, so we had a really significant gain into all synovial cell types. We created more than 100,000 single-cell RNA sequencing synovial profiles. And what we could see, we could identify 4 major populations. You can see them here: the T cells, fibroblasts, the monocyte macrophages, and the endothelial cells. And you can see also their proportions in the synovium color-coded in this graph. And then we had 6 minor synovial cell populations. And these were the plasma-situated dendritic cells, mast cells. Then there were B cells, and the pericytes and neural cells, and also plasmablasts. And because we worked with fresh synovium, we also could discover the small population of synovial neutrophils. And similar to AMP consortium findings, we could see really a significant interpatient variability of distribution of these major and minor cell types. We went further and performed the fine granulation, so to define the fine grade cell states among the bigger cell populations in synovial. So what we saw, we could define 9 macrophage, 3 dendritic cells, 7 synovial fibroblasts, 8 endothelial cells, and 8 T cell clusters. And similarly to the main cell types, we could also see a huge heterogeneity of these fine-grained synovial cell clusters across the patients. However, these were not the first insights into synovial tissue heterogeneity. The insights actually came from the analysis of the synovial tissue with the histology. And as you are probably, most of you aware, there are synovial tissue pathotypes. And these are different types of synovial tissue when you look at them with histology. And what we see, there is a post-immune or fibroid pathotype with the predominance of fibroblasts and other stromal cells, but lack of immune cells. And then we have diffuse myeloid pathotype where macrophages are predominating cell types. And further on, we have a B and T cell and myeloid cell dominated pathotype. And if you can now think, is that these different cell types might require different drugs to really affect and treat the disease underlying process. So what was interesting also was that the synovial pathotypes were shown to actually link to the synovial endotypes. So this means that these pathotypes were enriched on gene expression level for cell type specific gene modules. And these gene modules in different pathotypes could be actually linked to the clinical response and associated with RA prognosis. So the promyeloid inflammatory genes were linked to the response to initial therapy in RA patients, and plasma cell genes, so enrichment of tissue with plasma cells, was linked to poor prognosis and progressive structural damage. And then, of course, multi-omic-based analysis pushed the understanding of heterogeneity of synovial tissues further. And there was a study published on bioarchives that showed us that RA synovial tissues can be actually differentiated into 6 distinct cell type abundance phenotypes. So this is again a study from the AMP consortium, and what you can see here is that the authors collected RA and OA synovia, and what they did, they looked at the histology, they had clinical parameters for all of these patients, and they disaggregated the tissue for expanded single cell omics on all cells, all synovial cell types. And what they did two things. They looked at gene expression level, and at the same time, they also looked at the proteins that were bound to the surface of the, so the antibodies that were bound on the surface of the cells, and these antibodies identify the cell type. For example, CD19 would identify these cells. They took 58 different antibodies, and again, they created more than 300 single-cell profiles with both modalities, gene expression and surface protein expression. They found the main cell types, similarly to our study in fresh synovium, with the exception of neutrophils, because this is a frozen synovium. And they also could define 77 different fine-grained cell states across B cells, plasma cells, NK cells, myeloid cells, including dendritic cells and macrophages, and then stroma cells, including fibroblasts, pericytes, neural cells, and endothelial cells. And when they took these main cell types, you can see them here, what they could see actually was this clear stratification into 6 different cell type abundance phenotypes. For example, you can see here that EFM phenotype means enrichment of endothelial fibroblasts and myeloid cell types in the synovium. And what they further showed is that these specific CTAPs associate with disease-relevant cytokines, the histology, and also serology. They showed a significant heterogeneity of human synovium and also provided new pathobiology insights. So now that we got the significant understanding into synovial tissue composition and interpatient heterogeneity, I would just like to show you a few examples how omics technologies pushed forward the new breakthroughs in our understanding also of RA pathobiology and with potential for therapeutic innovation. So one of the interesting studies that use single-cell omics could show us that distinct fibroblast subsets drive different aspects of synovial pathobiology in arthritis, primarily inflammation and joint damage. What has been shown by the study authors is that synovium is populated by FAP alpha type 1 negative, FAP alpha positive destructive fibroblasts, which are located in the synovial lining, express matrix metalloproteinase, the gene signature, and in the sublining, we see FAP positive type positive immune effector fibroblasts. And these cells express and are enriched in inflammatory cytokine gene signature. And what the authors really elegantly did, based on these single-cell omic discoveries, they could sort the cells from the mouse synovial and adaptively transfer them to joints. And what they could see that if they transfer these destructive fibroblasts, they could see significant bone and cartilage damage, but very little impact on inflammation. In contrast, when they transferred the immune effector fibroblasts into the mouse joints, they could see significant acceleration of inflammation with minimal bone and cartilage damage. So this is an example how single-cell omics facilitated functional studies and also pathobiology discoveries in arthritis. What we know now is that different fibroblast subsets inhibit distinct synovial tissue niches, so the lining and sublining, and they drive different pathology, so damage versus inflammation. And this is now the space where we could probably look for new cell-based therapies that will specifically target each of these fibroblast subsets and address both pathologies in arthritis. Another very elegant study showed that resident macrophages create a protective barrier in the joint. And this was quite an unexpected finding. So what you can see here is the synovial lining and then synovial sublining. And what the author showed is that synovial lining macrophages actually make tight junctions. And these tight junctions are usually present in epithelial barriers, like in the gut. And tight junction formation between synovial macrophages now physically secludes the joint cavity from the interstitial sublining. What the authors also could show is that these protective lining macrophages restrict inflammation by, for example, removing dying neutrophils that are highly pro-inflammatory. Furthermore, in the synovial sublining, they could discover interstitial macrophages of two different subsets. And what they could show that a subset of MHC2 positive macrophages maintains the pools of protective barrier and the other interstitial resident macrophages in the synovium. So again, combining single cell omics and cell fate mapping and functional studies, we enable scientific discoveries and in arthritis and beyond. So this study really found out a very unique structural role of tissue-resident macrophages, and it diversified our knowledge about general roles of macrophages in health and disease. Finally, what the authors also could see in neuroarthritis is that this macrophage barrier that is tight in healthy joint becomes really disaggregated. It breaks during arthritis, and if you pharmacologically perturb this barrier, the arthritis in the mouse worsens. Still, the barrier macrophages during arthritis perform protective functions. And these data were then, that come from mouse arthritis, were aligned with human data. And what we know is that in active array, we could actually see this perturbed macrophage lining, and the lining macrophages are depleted. Furthermore, in remission array and in osteoarthritis, the lining structure is preserved and the lining macrophages are present. So if we look now on both studies, combining single cell omics and preclinical studies really can accelerate clinical translation. What we know now is that lining and sublining cell niches drive different aspects of joint pathology, and specifically targeting lining and sublining fibroblasts and macrophages and their subset could drive novel drug development for both inflammatory and degenerative arthritis. And finally, synovial tissue omics also provided a significant insight into stromal cell compartment, specifically endothelial cell fibroblast crosstalk in arthritis. So a few key breakthroughs in the last years. What we know now from mouse studies is that endothelial-mediated not signaling pathway regulates fibroblast identity. So it regulates differentiation of perivascular and sublining fibroblasts, and it also drives arthritis pathology. From human data, we know that these fibroblast links, notch gene signatures are upregulated in active and also in drug refractory array. And what was shown with the R4 array clinical trial is that enriched stromal gene signatures, fibroblasts and endothelial cell abundance are present in refractory array. So having this background, we looked in our team into endothelial cell diversity in inflammatory arthritis, collecting 9,000 single cell RNA sequencing synovial endothelial cell profiles, and we could identify an array of endothelial cell types spanning arterial, capillary, venous, lymphatic, and proliferating endothelial cell types. And this large body of endothelial cell types comes mainly from the venous cells. And similarly, as for other cell types, we could see really a significant variability of these endothelial cell clusters across different patients. And to look closer into the interactions of endothelial cells and fibroblasts based on the current knowledge, we started to build up the 3D microtissues, which you can see here. So these are a mixes of endothelial cells, synovial fibroblasts, and they are encapsulated in different hydrogels that represent extracellular matrix. And what we can see now, we can see the architecture of these 3D synovial microtissues, and you can see this nice development of the tubular endothelial cell structures. We also can dissociate this tissue to understand diversity of single cell states when endothelial cells and fibroblasts are mixed together or when fibroblasts are alone. And we also can perform the spatial cell omics. So looking how endothelial cells and fibroblasts interact on in a space. And for this, we can paraffin embed formalin fixed tissues, slice them, and put them on the specific slides, and then perform these spatial transcriptomics. And the spatial transcriptomics allows us to actually mount the tissue on the slide, and this slide contains such spots, and each spot is barcoded. And this barcode of the spot actually connects the location in the tissue with the gene expression in this location. So you can see now the mounted tissues that were composed of fibroblasts only and fibroblasts and endothelial cells together. We use the spatial transcriptomic technology from 10x Visium, and we could detect 5,000 to 6,000 median genes per spot. And what we did, we looked at each spot and deconvoluted gene expression of each spot. And what you can see here is that actually we could see 6 different clusters of transcriptomic variability. And this first cluster, based on the gene expression, identifies endothelial cells, and the other 5 clusters are more linked to mural cells and pericytes and fibroblasts. And when we look how these transcriptional cluster signatures actually are present or absent or enriched or depleted across the fibroblast-only tissues and fibroblast endothelial cells microtissues, you can see the presence nicely of endothelial cells only in the mixed tissues. And what is interesting actually that these specific fibroblast neural cell states become enriched specifically in fibroblast and endothelial cell microtissue. So the higher the yellow, the more expression. Furthermore, you can see that the strong fibroblast signature is really present in fibroblast-only tissue, but becomes depleted or decreased in the spots in mixed tissues. So what we can see was that the stromal cell signature differs in endothelial fibroblasts versus fibroblast microtissues. And looking at the genes that were enriched, we could basically see that basal lamina genes that are involved in the formation of basal lamina when the endothelial tubule is formed are promoted in mixed endothelial cell fibroblast tissues. And this raises the questions whether these fibroblasts that are in the proximity of endothelial cells can actually support the angiogenesis and formation of the tubules. And this is now the matter of further research. So with this, I'm coming to the summary. So what the single cell studies and breakthroughs in access to human synovial taught us is that we have now significant insights, and we changed our understanding of synovial biology fundamentally, both in health and arthritis. These discoveries have translational potential for development of novel cell-based therapies in inflammatory and degenerative arthritis. And finally, we better understand the synovial interpatient tissue heterogeneity. And when this is combined with discovery of novel pathogen-matched therapies, we hope towards accelerating the precision medicine gap closure in RA. And before coming to the acknowledgements, I just want to draw your attention to new research models and platforms that have been developed over the recent years. And these platforms and models can empower further scientific discoveries. One is our institute model, BioMed X, which is an independent research institute in Heidelberg, Germany. And we just opened a new institute in New Haven in US. BioMed X model of research has an academic research which is sponsored by industry partners. So this allows us really to focus on developing science at the interface of industry and academia. And the teams at BioMed X research in the fields of immunology, oncology, neuroscience, platform development, and artificial intelligence. There is a strong representation of different teams in autoimmunity and inflammation, looking into the leaky intestinal barrier in autoimmune diseases, looking into tissue protection, and also in dysfunction of T cells in autoimmunity and aging. And we just opened a new call in development of complex human ex vivo models of tissue inflammation in US. And this call closes in March 12. Finally, I would like to draw your attention to a new platform, Agorium, that empowers discovery through collaborative science. So Agorium empowers researchers to collaborate, share the data, and shine a spotlight on their own research in an efficient and accessible manner. And the key space of Agorium is to connect researchers and funders to facilitate collaborations, to innovate, and to accelerate access to the funding so that all of those three, connect, collaborate, and innovate, can become true. And with this, I'm coming to the acknowledgement slide. So again, I would really like to acknowledge all the team members of the João Enrico Fonseca Laboratory and Luis Grassa Laboratory at IMM. My team at BioMed X, our sponsor Johnson, our collaborators in Heidelberg University Hospital and at Friedrich Alexander University of Erlangen, and my previous team at University of Zurich, our collaboration with Robinson's lab in bioinformatics, the whole Center of Experimental Rheumatology, where I had my team before, and my very intimate collaboration with University Hospital in Ljubljana and University of Ljubljana, and all our sponsors. And thank you for your attention.
Well, Mojca, thank you very much for this very enlightening presentation. I have to underline that we have rheumatologists, we have rheumatology trainees, and we have some students here also in this room. So I think, and I hope this has been very interesting for everyone. So we will now open this talk for questions. And I forgot, we have also people from our lab, so please. Okay, so Mojca, one of the things that in a very subtle way you underline here is the importance of collaborating between centers. And this kind of research really needs that. You taught us about two kinds of ways to process the synovial tissue, to isolate cells, one from fresh tissue and one from frozen tissue. Can you please comment on the advantage of each one? Because at least from my perspective, for collaborating, we will benefit from the cryopreserved tissue.
Yes, so cryopreserved tissue the protocol was really developed to promote the retrospective analysis of the tissue and multicentric inclusion, combined with the single processing side, so that you avoid the batch effects. Of course, this gives an advantage of partnerships and retrospectively knowing the histology of the tissue and selecting which tissue can be processed further. And yeah, so this is the advantage. On the other side, processing fresh tissue can also be done in partnership. And I think this is one of the aspects of our partnerships. It's not that the tissue comes to you, but you come to the tissue and make a partnership. It can be made on a smaller scale, of course, but it allows really looking into every cell type, including, for example, synovial neutrophils that are very fast dying and would be lost during the freezing process. So there are pros and cons of both concepts, and yeah, it's coming to the tissue or sending the tissue.
Our mindset is also changing regarding that. The other aspect is some of these studies have very small numbers of patients. Of course, it's a very expensive technique, as far as we know. How reliable are the data and how reproducible they are based on small numbers of patients?
Yes, so what is basically happening now that we rely really on the smaller amounts of patients. There are limited amounts of accessibility to patients, specifically from the very defined clinical cohort. However, on the other side, also these very expensive omic technologies are getting cheaper and cheaper. And what we also know is that having access to these technologies, we can combine several modalities and we can also merge increasing the amounts of cells and decrease the cost. So increasing the amounts of cells can be still analyzed with the same amount of money as it was before. So this is one thing. And the other thing is, of course, that what we will find in this small cohort or smaller cohort, we can connect to a cheaper, maybe by cheaper ways of finding biomarkers, cheaper ways of finding the cell types. For example, with spatial targeted, looking at spatially targeted pathology of the tissue for a specific cell types that was found to be connected to a specific response, something like this. So I think we can translate these very expensive technologies into less expensive technologies and include more patients.
Again, any question?
Thank you, Mike. From the perspective of the guy who does some of the biopsies, sometimes we are not sure that we are getting synovial tissue. We are getting tissue around because in remission and in the even away patients, that may happen more often. How do you overcome that using these high-hand techniques? Because we may be studying some tissue around and not synovial tissue. So
yeah, so of course with this work on frozen synovium, you can always look in the histology, but you have to always rely that the pieces that were collected for the histology are equally representative of the pieces that were independently collected for, let's say, single cell omics. So, but the lottery is smaller compared to the prospective collection of the tissue. While you take the tissue, you analyze the tissue fresh, and only afterwards you can select which tissue qualifies as being synovium or not. From our experience in Zurich, the clinician had already some kind of insights, whether synovial tissue based on ultrasound picture, based on the look, physical look of the tissue, whether the tissue will be really highly likely synovium or not. So this was one. So we were always communicating this. And of course, we always had retrospective synovial biopsy on histology level and simply excluded the tissues that were not containing synovial pieces or representative synovial pieces on the histology. And these were out of the one thing that also happened to us is that when there was no synovium, we had no cells or very little cells, and we basically didn't proceed. So it was also that the tissues that were rich in cells and allowed us to do single cell omics were more or less always containing the synovium. And this was also achieved by the optimization of our protocol steps that really released most of the cells from the tissue. So we could really correlate the histology and the amount of cells seen in histology with the success to get the cells out.
Well, we're running out of time, so I'd like just to thank you very much for accepting the invitation from our group and just saying that we'll be together for a coffee break outside. Thank you. Thank you.